How is genetic information copied and transferred? Let us imagine the simplest case possible. The daughter cell must have exactly the same genetic information as the parent cell. For this to happen, whatever DNA is present in the parent cell must somehow be given or transferred to the daughter cell. To make an analogy, the daughter cell must receive what amounts to a Xerox copy of the parent cell's DNA. This is what happens when bacterial cells divide. But it is not as simple as Xeroxing the DNA. Let us look at the process in just a bit more detail. Just before the cell divides, the DNA double strand separates into single strands. After the double strand has separated, each single strand of DNA serves as a master or template for the copying or replication of DNA. The second strand is built up or synthesized based on this. Nucleotides are added, one at a time, following the sequence of bases found in the parent strand. The first base put in is adenine, and then the next, this time a thymine. The two are linked through a sugar phosphate bond. A third monomer, a guanine, is brought in, and the cycle begins again. The polynucleotide chain extends this way: cytosine, adenine, and adenine. This is repeated until the entire DNA molecule has been copied. The same process occurs with the other parent strand. Thus, after both strands have been replicated, the product DNA molecules are identical. The entire process is called the replication of DNA. Following the master plan or sequence contained in each parent strand, a complementary strand is built, one base at a time. Two DNA molecules, exactly the same, are the result. There is a point when the parent cell has two copies of the DNA molecule, but very soon after, the cell divides and each daughter cell receives one copy of DNA.